I am delighted to introduce our next keynote speaker, Dr. Boris Heifets. Dr. Heifets is Assistant Professor of Anesthesiology and Psychiatry and Behavioral Sciences at Stanford. He's also co-director of the Exploratory Therapeutics Lab. He is recognized for his work across both clinical research and basic neuroscience, integrating both human and animal studies. Dr. Heifets' research focuses on emerging rapid-acting psychiatric therapies such as ketamine, MDMA, and psilocybin that have the potential to produce lasting changes in system function, behavior, and therapeutic outcomes. On a personal note, it has been a very enriching experience to collaborate with Dr. Heifets on a Center of Excellence translational program in which we are investigating the mechanisms of ketamine and MDMA therapies. In addition to his research, Dr. Heifets is a board-certified neuroanesthesiologist, and he sees patients at Stanford Hospital, and he is also a scientific advisor at Osmind. Join me in welcoming Dr. Heifets, who will present on his advances in these exploratory therapeutics. And we will allow him more time.
I think this is my clicker. All right. Nice to see you all. Leanne, thank you so much. It is an enormous pleasure to be here. Again, part of my disclosure should be that I'm an anesthesiologist and not a psychiatrist. It is, again, been a great privilege to work with mentors in psychiatry, like Leanne, Alan Chasberg, Rob Malenka, Carl Deisroth, that have really brought me to be able to share the stage with them. So this has been an amazing journey. But I want to share some of our most recent work with you. Assuming I can get the clicker to... No? Oh, there we go. All right. So I'm going to tell you about a class of therapeutics that unless you live under a rock, if you've read the New York Times, Washington Post, NPR, you've probably heard about. Broadly classed as psychedelics. There's been a lot of organic, shall we say, groundswell of interest in this, decriminalization efforts. There's very little, I would say, known about how they work and actually even whether they work, but a lot of very promising preliminary data. So the first, really the prototype, and I'm grouping these drugs together because they have something in common. And this is one of the first real moves in this direction in the 21st century. This is ketamine. This is the first placebo-controlled trial of ketamine for major depressive disorder. What you're looking at here is mood rating scales, the Hamilton D, which many of you are probably familiar with, after a single infusion of ketamine, a 40-minute infusion of 0.5 milligram per kilogram. Now, we see similar size effects, even bigger size effects with psilocybin. Psilocybin is a classical psychedelic, a serotonin 2A agonist, among many other things that it does. This is a study from NYU, from the Ross group, again, looking at a depression scale, the HADS in this case. And you can see that there's an enormous separation after a single dose. Again, this is important that this is a small study. These are small studies. This was done in patients with terminal illness, looking at symptoms of anxiety and depression. And then finally, MDMA. So our lab works with all of these. I'm really going to talk about ketamine and psilocybin, but I would be remiss not to mention the last one here, which is the closest to FDA approval. It just passed a phase 3 trial, which was widely reported on and very successful by most metrics for the treatment of post-traumatic stress disorder. Again, what binds these therapeutics together is one dose, 1-2 doses maybe, and you have these weeks, days, months, weeks long effects. This is really not like any other type of therapy that we see in psychiatry or really in medicine outside of surgery, I'd argue. And so that's really what caught my attention. Is this plasticity? Is this some sort of catalytic event where you're allowing people to heal themselves in ways that they've never been able to do otherwise? That, I think, is one of the major questions, and that's really the argument that I want to present to you today is this is why we need to understand how these therapies work. They are racing through the regulatory approval pipeline. Again, MDMA is now 5 years. It's been on the breakthrough therapy designation, and psilocybin the year after, as ketamine, a version of ketamine has been approved now for a couple years. So these things they are coming. They will be very likely medically regulated within a couple years. But there's really a lot that we don't know. And again, these studies are very hopeful, they're very promising, but they're small. By any other metric, if you were to compare this to a cholesterol drug or a hypertension drug, there's a long way to go, and there are a lot of things we do not understand. MDMA most certainly has abuse liability. Same can be said for ketamine and S-ketamine. Psilocybin, again, there have been so few patients treated worldwide in any kind of controlled setting that we just don't know who benefits and under what type of depression, or is it even depression, or is it something else that we're treating? So this is why I think we need to understand mechanism. And one of the biggest questions facing psychedelic therapy is it the drug or is it the experience? And that's driven by this, very widely reported experience that people have these transformative experiences under the influence of a drug, and that is not like the model for SSRIs, for example. So what is that? What is that telling us? Like, how do we get at that mechanistically? And it raises issues for doing double-blind trials. And really applying the standard methods that we've gotten so accustomed to understanding how therapeutics work. So I want to share with you 3 stories from our lab and our broader research group that get at this in different ways. So is it the drug of the trip? I mean, I should just say at the outset, I have no idea, and if you leave here thinking that you have an answer to that, then I have not done my job. There's a lot that we don't understand. The first thing I'll tell you about is a preclinical study. And this is looking at the idea of set. So set and setting with classical psychedelics is something that has been in the zeitgeist for many years now in this area. And it really gets at this idea of how does the environment you're in interact with the drug that you're taking and the experience that you have and the therapeutic benefit that you might achieve. So we decided to get at this question in mice where we have a lot more control, and you may wonder what mice have to say about psilocybin, but I will hopefully convince you that they have something to say. The next way that the approach that we've taken to answering these questions is in a human trial. So ketamine is probably one of the best studied among this loose class of drugs. And there's been a lot of discussion about the dissociative component. How much does it matter how much you dissociate, how long you dissociate? Do you even need to know that you dissociated? Is that really an important element? So being an anesthesiologist, everything looks like a nail when what you have is a hammer. So what we did is we gave ketamine during surgical anesthesia. Patients they don't, by definition, have that experiential component, yet they're still getting the drug. They're still getting all of the trappings of the therapy. And then finally, this you'll see, but this will lead right into this idea of, psychedelic-like states without psychedelics. Should we be thinking more about this as a physiological process, a set of circuits, let's say, that we can bring online to achieve a therapeutic transformation rather than a receptor-specific activity that has to happen in order for therapy to go forward? And that's going to be a case report, which I'll get to in a little bit more detail. So set and setting and psilocybin. So it's been known for a long time, going back to the days of Timothy Leary, that the setting in which a drug is taken can influence short and long-term effects of psilocybin. And what you see on top there is from Matt Johnson at Hopkins, the site of the world's first psychedelic research center. And that's the couch, that's the lighting, that's the therapeutic environment. You put on eye shades, headphones, you take a drug, you have an experience. And that is believed to promote better therapeutic results. And yet, this is a survey study from Molly Crockett at Yale, down below. I don't know which festival it's at, but reported therapeutic effects may occur in different settings. So people in all kinds of naturalistic settings, things that we don't typically associate with therapy, can also be therapeutic. So which one is it? Does the setting actually change the effect of psilocybin on brain activity? And this is something that we can answer in mice. We don't have to make any assumptions about behavior. What we're looking at is just brain activity read out by immediate early genes, which is a good marker for physiology. So what we're trying to get at is, do drug and setting truly interact? Does the effect of the setting change the effect of the drug on brain activity? or is it just additive? You get therapy, which can be great, you get psilocybin, which may help, and you put them together, and are they really a bundle? So this is set and setting for the C57 Black 6 mouse as best as we could. We gave mice a dose of psilocybin or normal saline, which is just a vehicle. And we either sent them back to their home cage, which I'm not going to say that this is therapy, but maybe it's therapy. This is more of a comfortable environment. It's not too disruptive, it's social. Or we sent them to an enriched environment, again, group housed. This is as close as we get to a mouse rave. This is, again, the enriched environment is something that's well studied in many other contexts. It promotes neuroplasticity, learning, memory, resilience in stress models. So these are two powerful interventions. The enriched environment intervention in any number of mouse studies has a lot of it's a big thing to do behaviorally. Psilocybin also, has, as humans, any human can tell you who's taken it, also is a big intervention. So what are we doing? We're trying to do this in as unbiased a way as possible. We're not looking at a specific behavior, we're looking at environment. We're not looking at a specific brain area, we're looking at the entire brain. How do we do that? We randomize these mice, we remove their brains, we clear them, we do some very, very fancy imaging, which you can see there, and if I had there we go. I could look at these all day. This is what these little dots of light are actually cells that were active using a marker called CFOS during the drug experience. And again, using at great, with a lot of sweat and tears, this is work by Austin Casey, Dan Rusketich, and Dan Barbosa, who developed this pipeline. With a little bit of machine learning, we're able to categorize all the cells across the brain, count them up, and then parcel them out to each of their individual brain areas. So what is it that we're looking at? The marker is called CFOS. I'm not going to dwell on this, but basically this is a pretty reliable marker, an echo of neural activity. And what you see here is just the number of action potentials scales with the amount of CFOS that you express. So again, after we take these beautiful images, we register them to standard atlases, and then we get these statistical maps of activity. And this part is important, I think, in that what we're able to do is we're able to build maps that really that highlight certain contrasts. Psilocybin versus saline across all contexts. Adding all the noise of enriched environment or home cage, we put them together, and then we look and see what survives, what's the drug-specific effect. We do the same thing with context. And then the real heart of it is that drug-context interaction. At any point, for any brain area, does psilocybin actually, or does the novel enriched environment change the effect of the drug? That's the question that we're trying to set up here. So this is psilocybin drives an enormous effect in CFOS expression. What you see in the spinning brains on the left hand is where psilocybin actually suppresses activity. This is a network of subcortical regions. Where you're looking at on the left hand side is the Allen Brain Atlas, which is basically kind of a color map of cortical and subcortical areas, and you can see from the sunburst plots where these clusters tend to reside. On the right, as again has been shown in numerous imaging studies, psilocybin tends to enhance cortical cluster activity. Great, no really big surprises here. What we're able to do is to actually validate this kind of broad expression at cellular resolution. And this is really, we were helped along by Zhui Zhang and Lian Williams' lab to do some of this analysis. But what I'm showing you here is we're able to actually isolate 30 different clusters validated at the single cell level across the entire brain. Now think about this in terms of what we could do if we could do this with human imaging. But again, these are associated with acute psilocybin independent of context. Most of them, very large clusters went up, 8 went up, and 22 smaller clusters went down. And the overall picture is that of enhanced neocortical activity and suppressed subcortical activity. This is the effect of context, right? So we have two big manipulations, drug, environment. This is what they look like independently when we look across all conditions. And what we take away from this is that, yes, indeed, these are two large manipulations on brain activity. And again, we're able to identify a number of clusters, and I'm not going to go deeply into, but we have a broad network of cortical and subcortical clusters that are responsive to enriched environments. And this is, again, grouping together saline, psilocybin, everything. Now, the question that I was most excited about is, what about the interaction? So we were surprised that despite the enormous effect sizes for psilocybin and enriched activity, there was actually no clear interaction. We even dropped all of our normal statistical safeguards and looked for, and drop our false discovery rate to look for even a faint signal. And what we see is just maybe a couple regions, which again are enticing, but this really, it does not stand up to any sort of statistical rigor. So what we take away from this is that there is a massive effective drug, there is a massive effective environment, but do they really truly interact? Again, it's hard to, I mean, we want to anthropomorphize it, but again, the kinds of questions that this leads to for human studies are, maybe these are really, this is telling us that these are separable effects. Having good therapy is helpful. Maybe having psilocybin is independently helpful. Is it really, how much does it really matter what you do? It matters to the extent that you're delivering good therapy. Maybe that's really what we should be looking at. And so how do you get at that? In human trials, we'll get at that in a minute. So again, just to sum up what we believe we know, which is that both of these manipulations have massive independent effects. We're using a marker that reflects neuronal activity, and this is as unbiased as we can. No specific behavior, just broad environmental manipulations, and again, looking across the entire brain. And we are very much looking forward to our continued collaborations with Leanne, Carl, Tricia, others to do this sort of work in humans and validate some of these approaches using human imaging. So now getting on to humans, we can actually answer this question in a population of convenience. I work in the OR a day a week. Millions of ketamine anesthetics are given across the country every year. Why is it the anesthesiologists were so slow on the uptake to recognize a mood effect? And there are a lot of potential answers to that, but there are very few rigorous systematic efforts to do a psychiatry trial in the operating room. So I'm going to give you a little bit of background on why, again, we should care about ketamine. Ketamine was developed over 50 years ago as a dissociative anesthetic. This is from Corson and Ed Domino, who passed away last year. And again, this is that same, it took 40 years until we got the first clear evidence of efficacy for treatment-resistant depression. In the intervening years, there's been, I have to say, breathless coverage by much of the media on the wonder drug of ketamine. That's evolved to companies like MindBloom, where you can take ketamine at home. There's very little evidence around these practices. This is from a company called WonderMed, where you can take low-dose oral ketamine by mail order. So I don't know how safe that is. I don't think anyone knows how safe that is. And this should be a concern, and I'll tell you why. This is a meme on Reddit I found in 2021. I had to look hard for this. This is what people think about, what they talk about ketamine. In 2022, some of my favorites is ketamine the best drug in existence, self-medication. I tried it for the first time from a stranger on a train, and my favorite, what do you do for fun? Oh, I like to do ketamine in my room by myself and watch movies. So this is a potential issue. There's an abuse liability here that widespread, broad dissemination of ketamine is not necessarily in the public health interest. So where is this going? Well, this is a dispatch from our colleagues to the east in Hong Kong. Hong Kong is fast becoming a K society. K is in ketamine. As a result, about 80% of young drug addicts, their words not mine, are taking ketamine. So much so that in 2015, the People's Republic of China tried to get it put on Schedule 1, which is the most restrictive category of drugs. So this suggests that they had a big problem with ketamine diversion, illicit use, abuse. And again, this is something that we do not want to go in this direction. So now that I've scared you, what do we know about the benefits? This is a study that we did in collaboration with Osmind. The lead author is Tuli Hedemius, who showed basically, again, not a surprise, we've seen this before, ketamine has massive treatment effects in real-world settings on depression and anxiety. Now, there's a problem here. Again, all of the things with placebo, dissociation, how much of this is just regression to the mean? So with Leanne's help, we did, I think, a really cool thing. We looked with 2 control data sets in people who hadn't gotten ketamine in some of these same settings. The iSpot data set where people started taking SSRIs, SNRIs, and in the 2 weeks after they start, before there's a clear biological treatment effect, where all of that expectancy plays in, that's basically, we can estimate that effect on the top 2 lines. And those are remarkably similar to non-treatment controls. That's the expectancy, the non-specific portion. And the big, what we were very excited to see is that after a series of ketamine infusions, we're still getting a potential benefit of ketamine in real-world settings. So there's some sort of public or large N control that we can apply. This still doesn't really get at the idea of placebo effect and unblinding and the interaction with ketamine. So how important is dissociation actually for that antidepressant effect? So there's some work done from the NIH group, again, looking at there is a relationship, it's not strong, between how much people dissociate on drug. That's really what people often now term as the psychedelic-like effect. There's a relationship between that and how much they get a therapeutic benefit. The real question is, how do you control for it? How do you do an active placebo that mimics that dissociative effect where people don't know what they got? What you can see here is a study, again, from, I think, the NIH group, where looking at midazolam as an active control versus ketamine. And you can see that, again, the treatment effect for ketamine is about 50%, 60% response rate. Midazolam has a non-trivial effect, and again, some of this has got to be due towards functional unblinding on drug. So what's in a placebo effect? And this is, I think, very important. We should not, placebo is not just something that we fool our children with and discard in clinical trials. There's actually something very therapeutic that we're just beginning to understand the mechanisms for. So again, how does it play into it? Positive expectations, obviously, have an effect on outcome. Negative expectations also interfere when you know you did not get the active treatment. Setting expectations is critically important. That's setting preparatory psychotherapy. And this is one of my favorite studies. Again, there's placebo circuitry that we know something about. This is remifentanil, a potent opioid, and pain responses when you have at baseline given no expectation of what the therapeutic effect is going to be, a positive expectation, and a negative expectation. You can completely ablate the effect of remifentanil on pain perception by setting expectations. That doesn't mean that we discard this trial as hopelessly confounded. It means that there's a real mechanism behind placebo, and perhaps we should be exploiting it. And how do we actually go ahead and do that? So this is basically the question that we're trying to ask by putting people to sleep during surgery and giving them ketamine. We have 2 hours plus of where we do detailed histories, do the mini, do these the scores. They have a drug session where now they're no longer aware. They're all, I promise you, they're all fully asleep for a variety of surgeries, not cardiac, not neurosurgery. And then we follow them up closely. This is really the model for a lot of, for psilocybin, for MDMA, and for some forms of ketamine therapy. So what did we do? We tried, again, this is with help from Alan Schasberg, Laura Hack here. We tried to make this look as much like a psychiatry trial done in the OR as we could. We took moderate to severely depressed patients that were there for non-cardiac, not neurosurgery. You can see here, I'm not going to go through this in detail, but they skewed, they were around 50, skewed female, and they were typically 20 years duration with some degree of treatment resistance. So we look at this and we say, this is reasonably comparable to the trials that have come before. And what do we do for outcomes? We use all the clinician-rated scales, all of the secondary scales that other people use. So what happened? All right, as placebo. So this is why I spent so much, I really want to drive home what is going on in this placebo group. This is probably one of the only, if not the only, trial of a psychedelic adjacent medicine where we actually test the blind and have a perfectly blinded trial. What does ketamine do? Not much more. So we get a gold star for having some statistical separation that is clinically insignificant and has more to do with baseline characteristics than anything else. This is not, we believe, due to low power. But there are substantial improvements in both groups. These are, again, patients with a degree of treatment resistance with moderate to severe symptoms of depression. If you look at those baselines, they're not too far apart from what has come before. So what is this telling us? Again, let's look at the response rate. The response rate for both groups hover around 50%. The remission rate is about 30%. Like, these are really, if I didn't tell you that these were placebo controls, you would say, what a promising new exciting treatment for treatment-resistant depression. This is just a comparison from 2013. Again, that same study I showed before, showing very similar response rates for ketamine. So again, it's comparable to previous studies, and it really suggests that placebo, maybe it's more effective during anesthesia because we don't have that issue of unblinding. So let's look at the data another way. This is, again, grouped by treatment allocation. This is grouped by the patient guess. So you can see here, patients who believed I got ketamine, they did a lot better. This, again, demonstrates, again, you can see the numbers there. We got a really good blinding result. And then everyone was 50% in each group, but patients who believe they got ketamine, they had an expectation that based on their response, they must have gotten ketamine because ketamine works, right? So what I take away from this is not that placebo is something to be discarded, but it is an incredibly powerful intervention when you don't disabuse people of the notion that they got placebo or they got ketamine, that they don't know what happened during surgery, that you can get very powerful treatment effects, and we should be finding ways to leverage this rather than maybe to discard it. And this, I just want to read very, very quickly one account from a patient in the placebo group who thought she got ketamine. She had a very difficult course, and I just want to read the last part. I was taking 6 college classes while waiting for surgery. I was able to take all 6 exams from the hospital bed, pass them all with A's and B's. Someone who went taken back for near paralytic complications of an implantable cervical spine nerve stimulator for chronic pain for a complex regional pain syndrome. These are sick patients with a lot of comorbidities. They're still able to get these massive experiential changes. So this brings me to the last set, and I see the yellow light, so I'm going to make this really fast. Can other non-ordinary states of consciousness be therapeutic? This is a case report from the OR. This is one of the coolest things I've seen in a decade, led by Harrison Chow, who's a staff anesthesiologist here. This is a woman who had a knife attack 12 days prior to us seeing her. 12 days of nightmares, sleeplessness. Laura Hack, again, psychiatrist that we've been working with for quite some time now, diagnosed her with acute stress disorder, something that would probably go on to post-traumatic stress disorder. And she came to us for local anesthesia, or regional anesthesia for her arm, and monitored anesthetic care, which is basically sedation. What you see there is the blood-brain barrier where the surgeons work, and then the patient is gently asleep, deeply sedated for the procedure. So I'm just going to walk you through this without, again, broad strokes here. Patient was sedated. What you see in purple, that line there is that is the patient state index. That's how asleep they are. And what you can see is at some point the EEG transitions to something REM-like, and then we identify something that looks like a dream. And what happened next was really remarkable. The patient wakes up, tachycardic, and recalls this dream where she relived the attack. She remembered presenting to the emergency room, going to the OR, and then remembered in her dream going home, using her repaired hand, running errands with total resolution of her symptoms in her dream. And she wakes up with complete remission. We followed her out for 6 months. Something that should have progressed to PTSD, potentially, is able to resolve with a single powerful experience with a dream, an anesthetic stabilized dream. That, again, this is an N of 1, but it suggests the many shapes that powerful, transformative psychological experiences can take. And again, this is something that we're intensely interested in. And we can develop technology around this, but again, it drives home the point of physiology over necessarily pharmacology, and that we really want to achieve a therapeutic state, and how we get there we're just beginning to understand. So this is the last point, is that drug mechanism matters, how these drugs work, how the therapies, the complex bundle of drug-assisted psychotherapy is critically important for us to understand. We need rigorous clinical science, and again, physiology may be more important than just a receptor. I want to thank a lot of people who I am not going to name in detail, but a lot of mentorship and a lot of help from many departments, particularly psychiatry, across the university. So thank you very much for your attention.
Thank you, Boris. Do you want to take questions, or you're okay with people taking them in the break?
Yeah, either way.
Either way. Okay. Thank you for a really incredible presentation and the provocative ideas. We're going to do a mix. If you feel very hungry, lunch is ready. It's outside. You can get it, bring it back in, or eat outside. Dr. Heifetz is willing to answer questions for those of you who'd like to stay on and cut into the lunch break. So we can do a bit of a mix. Do we have any questions from the floor? If not, definitely Rachel has one.
Lunch calls.
Okay, so we have a question. For the separation of ketamine and placebo response, is it possible for the placebo treatment effects, or both, are actually due to altered states from the anesthetic?
So that's an excellent question, and I think I'm going to boil it down to, do anesthetics themselves have antidepressant responses or antidepressant effects? And again, I don't want to split, but are they biological antidepressant effects? The answer to that, I think, is a probably not. This has been looked at by a number of groups, notably Utah, Brian Mickey's group, Peter Nagle, looking at various anesthetics. Nitrous on its own, nitrous oxide may have some antidepressant effects. That's undergone a phase 2. None of these patients got nitrous. That was actually an exclusion in this case. Studies with propofol use very high doses, very repeated doses, that drive patients into basically electrical silence, called burst suppression, EEG. That was not the case here. So it's unlikely, it's possible, but it's unlikely that the state itself caused by anesthetics was somehow biologically antidepressant in terms of some specific effect on dendritogenesis or something like that. Much more likely it was a kind of experiential interaction between preparation, expectation, giving up some control prior to surgery, and then close follow-up that drove those outcomes.
I think people are looking for food, so thank you again for a fantastic presentation. Lunch is outside, so please make your way there. We'll start again at 1 p.m. with our next plenary speaker, Carl Diceroth.